All right, so for the last talk of this session, we're going to hear from Hamutal Arbel, who's going to tell us about an accurate method to discover a subset of enhancers in the early blastoderm.
Hi. Hi, my name is Tali. I'd like to discuss a method that we found to be very accurate in predicting a subset of enhancers in stage 5 Drosophila embryos. I'd like to touch briefly on the motivation, the data that we collected, and how looking for a subset of enhancers that are related to a particular biological role instead of looking for the entire set of enhancers everywhere really improved our ability to predict enhancers. I'll touch briefly about what we think these enhancers are doing and what we have done to find these enhancers genome-wide and what we're doing to validate our results. Finding enhancers has really been one of the fundamental questions in biology, and there's been a lot of research focused on this. We've heard several attempts today. A lot of data has been collected, and a lot of attempts have been done at the genomic level, and also a lot of attempts have been done computationally using whole genome data, genomic data such as ChIP-seq, ChIP, and StarChIP. And although there have been many attempts and there have been many publications, generally speaking, the false positive rates have just been too high to make very useful whole genome scans. For instance, the ENCODE consortium segmented the entire genome into functional annotation, including enhancers, weak enhancers, and non-enhancers, primarily based on ChIP-seq histone modification data. But when an independent validation was attempted by Barak Cohen's group, it was found that only 24% of the enhancers and weak enhancers showed more greater expression than their scrambled counterparts, which corresponds to a false positive rate of about 70 to 80%, which was high. So clearly there's some room for improvements there. We could also note that weak enhancers actually had more expression than the enhancers. So clearly there's some biology there that we just don't understand. So why is it that such top-of-the-art excellent methods fail so completely? We believe that at least partially this is because people are just trying to do too much, trying to find all enhancers everywhere instead of accounting for the biology of the problem. So we have focused our attention on the early Drosophila embryo at stage 5. The reason we're focusing there is because the transcription regulation network is much simpler. It is very well known. And specifically, we have been focusing on patterning, which only has 42 non-transcription factors in its network. So we have a very good probability of hitting the major players in the system. And of course, there are all the usual reasons that make Drosophila such a wonderful model organism, and also the fact that it is very easy to image, particularly at stage 5, when all the cells are at the periphery of the embryo. As our training set, we used the KVON data set, where they have produced a very large number of GAL4 receptor assays. They inserted the genomic region proximally to GAL4, inserted it into embryos, and then collected overnight plates. So we had embryos of various stages, hopefully all stages, then hybridized, fixed, imaged, and annotated. However, while this was brilliant work, and they did 7,705 of them, which gives me a lot of data to work with, this was still had some of the problem of very high throughput experiments, where not all plates had embryos of the relevant stage, and there were few embryos imaged at some plates. We also had 282 regions tested at very high quality by Sus Selniker group, and those also had a high number of enhancers. Altogether, I had close to 8,000 enhancer regions, DNA segments, out of which 800 were enhancers at stage 5. Our variables were 43 ChIP-seq data for histone modifications by the Eisen group, who also did the ZELDA transcription factor. We had ChIP-chip data for 21 transcription factors involved in patterning, which is half of all transcription factors involved in patterning at stage 5, which was really quite a lot for us. That was thanks to the Berkeley Drosophila Genome Project. And with all this data, and we used all this data to fit random forest, which is a supervised machine learning algorithm. And with all this data and everything that we had, we got results that were very, very mediocre. We had the area under the ROC curve of 0.83, which is comparable to what the ENCODE consortium got, and it again will give us about 70 to 80% false positive. However, if you note, the ROC curve actually has a sharp rise at the beginning, and this is because there is a subgroup of enhancers which are correctly classified again and again and again and again. These are homogeneous subclass, and if we set all the other enhancers to 0, say they are not part of our training set anymore, and only focus on those particular enhancers, our predictions become drastically more accurate. Our errors drop by a factor of 5, and this really indicates that there is a boundary between the different enhancer groups. With this set, we can predict more than 98% of the enhancers with an accuracy greater than 95% when there is an equal number of enhancers and non-enhancers in the test set, which definitely is good enough for a whole genome scan. These enhancers are not random. We find that these enhancers that we can effectively predict drive expression in the primordia of specific body patterns. We also note that if we do GO-term analysis of proximal genes, we see that these genes are very much enriched in segmentation terms, such as blastoderm segmentation, pattern specification process, etc. So we believe that what we have found here is a subcategory, segmentation-driving enhancers. These enhancers are homogeneous among themselves, and they are much closer to each other than they are not only to non-enhancers, but to other set of enhancers, which you can see separate beautifully from them. Now, they separate across the expected bicoid transcription factors, which has to do with segmentation. That is not surprising. But they also separate completely from other enhancers in ZELDA, which is a more general transcription factors. And most surprisingly for us, they are also completely separated in histone modification marks, which indicates to us, at least partly, why it may have been so difficult to find enhancers. And when trying to find enhancers based on histone modification, it is assumed that the histone modification is going to be the same for all sets, if you're trying to find all sets. And we find here that if we're in different sets of enhancers, there is a difference in expression. In the whole genome scan, we find that more than 80% of the entire genome has 0% probability of being an enhancer. It has no probability at all. However, at the very top of the probability histogram, there is a secondary bump. There is a second. This is actually a bimodal distribution. The area between between 0.75 and 1 holds 1.6% of the genome, and this is where we believe we can find these segmentation-driving enhancers. Altogether, we predict 1,600 segmentation-driving enhancers. More than 1,000 of them are novel, meaning that they are not in our predictive set, and they are not in red fly. And we rediscovered de novo 98% of our training data. For validation, Sue Selinkler is making a transgenic flight holding 50 of our top enhancers. But while these results are not yet in, we have gone back to look at our training set and looked back at our false positive. That is, those regions that our model predicts should be enhancers, but the experimental results said are not. And we find that in most cases, there either aren't any embryos of the relevant age on the plate, or they are enhancers that somehow fell through the cracks. And you can see 4 examples of such next to you. And when we asked an independent researcher to look at our top 100 false positives, she could only find 16 where she could confidently say those were not enhancers. So, to summarize, enhancers are heterogeneous. They are driven by different biological systems, and it makes sense that they will respond to different biological cues. Looking for all enhancers may be difficult if they don't all respond to the same things, and there may be different rules. However, if we account for the biology of the problem, we can make very accurate predictions. This is mostly driven by the transcription factors, so we definitely need more transcription factor studies to get better understanding of other biological systems. And with that, we can use random forests to find homogeneous subgroups in the data. Thank you. And thanks, Ben. So I want to thank Ben Brown, my advisor, for mentoring me, Peter Bickel, Sue Selinkler for hosting me at the LBNL, Mark Biggin, the head of the consortium, Solly and Samantha for all the help in the project, and all the rest of the group. Thanks.
Okay, questions?
Thanks for that nice talk. Can you tell us one more time over here?
Oh,
Hi.
sorry.
Can you tell us one more time what the step was that got you from the lousy area under the curve to the fantastic area under the curve algorithm?
So,
you just focused on the top ones, the top scoring with your machine learning vector, and just
random forest.
Yeah.
Similar. I can't go back. Okay, sorry. So the initial data contained 798 different regions, enhancers. That's all the enhancers we had for stage 5. And then we did an initial study, and we saw which one of them coalesced together we could predict again and again. We ended up with a group of 358 enhancers that had more than 75% to be correctly classified. And then we set the rest of them to 0. We said these are not enhancers, they're not part of our prediction set. And we only predicted with those that we could, that were working. Yes. Thank you.
So you were validating the false positives, or what you originally called false positives, by actually doing enhancer assays. But what if you just ask what the nearest gene is?
We looked at the nearest gene. We did some analysis of that. Do you mean for the enhancers themselves?
I mean for the ones that you're calling false positives.
Well, we did that analysis. I don't remember that we got anything very significant. Thank you.
Okay, so if there are no more questions, I would like to thank all speakers for excellent talks today.